Thank you very much. Good morning, everyone. I have the pleasure of introducing our first undergrad speaker for our summer speaker series. Super exciting. So I wanted to introduce Abigail. Abigail Anderson, she comes from Chipley, Florida, and she will receive her bachelor's degree in behavioral and cognitive neuroscience with a minor in philosophy in 2023 from UF. After graduation, she plans to pursue either an MD or a PhD, but she is still undecided. She's currently in her fourth year doing research in the lab of Dr. Malu Tanzi at the McKnight Brain Institute. The Tanzi lab focuses on investigating the role and regulation of neuroinflammatory and immune system responses in modulating the gene-environment interactions that determine risk for development and progression of neurodegenerative and neuropsychiatric disease. Okay.
We'll break that
down. So we are really excited to hear your talk. Thank you so much.
Thank you, guys.
Maybe pull the mic just a little closer to your mouth. Go ahead, ready to share?
Can you hear me? Yes, yeah, if you can put that up there. All right. Well, thank you for that introduction, and thank you guys so much for inviting me here. I'm really, really excited to give this talk. Any opportunity I have to talk about my science, I love to do it. This is like my bread and butter. This is what I spend all my time doing. So I'm here today to talk about neuroscience. That's what I study. It's what I'm absolutely in love with. So we'll break it down a little bit, what she just introduced, and how that ties into what I study, what my lab studies, all of those things. So my presentation is titled, Loss of Progranulin Results in Increased Pan-cathepsin Activity. And I'm going to define all of that so we know what we're talking about. But to start with, I want to talk about what my lab focuses on. So I'm in the lab of Dr. Malu Tanzi, and I was really fortunate to start at UF the same year that she started at UF. So it was my freshman year. She just got recruited by UF from Emory to start doing research here, and our paths crossed, and I fell in love with what her lab does. We matched, and I joined in her lab in the fall of 2019, so fall of my freshman year. And she has taught me absolutely everything I know about neuroscience. I thought I loved it before, but I know that I love it now. And so what this lab focuses on is preventing or delaying neurological disorders instead of trying to treat them with therapeutics. So a lot of research has showed that neurological disorders like Parkinson's disease, people start showing symptoms 20, 30 years before they're actually diagnosed with Parkinson's. So how can we come to understand these symptoms and address the problem at that time so it doesn't lead to the full development of this neurological disorder? And another sort of term for neurological disorders that I'll use is neurodegenerative diseases. And this is really just when the cells of the central nervous system stop working and die. These get worse over time. They don't have a cure. And that's why it's so important to be doing research in this field right now. Examples of neurodegenerative disorders you may have heard of include Alzheimer's, Huntington's disease, even Lewy body disease, and of course, Parkinson's disease, which I'll talk a little bit about today. And so the way that my lab sort of focuses on these is that we think that the immune system works to keep the brain healthy throughout a lifetime. So the immune system and the brain are partners. And over time, basically as you age, you start to undergo chronic inflammation. So you have really, really low inflammation when you're young, and as you age, this inflammation tends to get worse. And inflammation is an immune response, so throughout your lifetime, you experience varying levels of inflammation, but there is this chronic inflammation we refer to as inflammaging. That's really, really important. So we think that the brain and this concept of inflammaging are tied together and lead to some of these neurodegenerative diseases. So the big question that my lab is trying to answer is, how does the immune system protect against neurological disorders? And we really want to try and understand the biological mechanisms behind the immune system and how the immune system might be a first responder to neurological dysfunction. So because of that, my lab is doing so many different projects. They're focused on so many different molecules and all sorts of things, but my very small piece of that is progranulin. Before I get to progranulin, I want to talk about these 3 neurodegenerative diseases because they're very important specifically to what I study. So the first one would be frontotemporal dementia. This is caused by degeneration of the frontal and temporal lobes of the brain, hence the name. And symptoms include language problems, movement disorders, even personality changes. And these are common symptoms you'll see across a lot of different neurodegenerative disorders. Another neurodegenerative disorder that's relevant to progranulin is neuronal ceroid lipofuscinosis. So this term actually covers a group of lysosome storage disorders. And lysosome storage disorders are that's just what it's called when there's excessive storage of cellular materials done by the lysosome. So specifically NCL11 is relevant to progranulin. Type 11 is characterized by visual loss, seizures, uncoordinated movement, symptoms such as that. And this is caused by the loss of heterozygosity, so loss of both alleles of granulin. And I believe I skipped over that talking about FTD. So FTD is caused by a haploinsufficiency of the granulin gene, so loss of one allele. And then we have Parkinson's disease, which you all may be a little bit more familiar. It's talked about a lot more. And this is characterized by tremors, muscle rigidity, and difficulty with coordinated movement. So this is 3 different neurodegenerative disorders. It doesn't make sense that I would be studying all of these. That's a really big span to cover. But there's something that ties all of these together, and that's progranulin protein. And that's what I'm really here to talk about today. That's the molecule that I study. I spend all my time looking at progranulin protein. So what exactly is progranulin? So it's a secreted glycoprotein. A glycoprotein is a protein plus a carbohydrate, so a protein with a sugar. And it can be processed into granulin peptides in extracellular space. So granulin peptides are just an active part of progranulin that's broken down. So progranulin is encoded by the gene granulin, and that's why we talked about the haploinsufficiency and the heterozygosity of this gene granulin that leads to some of these neurodegenerative disorders. So it's encoded by this gene granulin. And what we know right now is that progranulin protein is really important for cell growth, survival, and repair. And we know also that granulin peptides often act opposite of whole progranulin. So you have whole progranulin that does one thing, and then when it's broken down into these granulin peptides, it does something opposite. And an example of this is progranulin has an anti-inflammatory role, but granulins, once progranulin's been broken down, seem to have a pro-inflammatory role. The other thing that's really important about progranulin is that it's localized to the lysosome, and it's highly expressed in peripheral immune cells, such as monocytes and microglia. And so that's what I'm showing in the image on the right up there. That was not taken in my lab, but at the top, those are results from a Western blot, and I'll explain what a Western blot is in a little while here. But you'll see on the top, that's progranulin, and this lab looked at the levels of progranulin in neurons compared to microglia. So you'll see in the microglia lane, there's this really dark black band. That means the progranulin levels were basically through the roof in this microglia, and there's almost nothing visible in the neuron. Oh, perfect. There you go. That's what I'm talking about. So in the neuron, there's basically nothing there. Microglia, it's super, super concentrated. So it's just important to know that progranulin, that's the type of cells that we're focused on here, are microglia. And then there are 2 pathways that progranulin uses to travel into the lysosome of the cell. This is sort of just background information. It's not super important to what I do, but just as sort of a side note, progranulin can bind to sortilin. This is a trafficking receptor, and just be delivered directly into the lysosome. Or it can bind to PSAP, which is a soluble lysosomal protein, and then go on to bind to CLM6PR or LRP1, which are other trafficking receptors, which will then carry it into the lysosome. So those are the 2 main ways that progranulin can get into the lysosome. So the other important thing about progranulin is any variation in expression of progranulin causes some type of problem. So there's sort of this like perfect level we want to be at, and as soon as we change it, really weird things start happening. So if there's underexpression of progranulin, where this is what's been seen as a risk factor in AD and PD and ALS, and it's a risk factor in schizophrenia, bipolar, and other psychiatric disorders. Low progranulin seems to worsen tissue damage. It also promotes neuroinflammation, which is one of the big things that I'm worried about. It also enhances peripheral inflammatory conditions like arthritis. And then high progranulin on the other end of the spectrum, it can promote cancers such as multiple myelomas. It can promote carcinomas of the ovary, liver, breast, kidneys, and even prostate. It also can promote brain cancers, regrowth of injured peripheral neurons, and also increase neuronal cell survival, neurite extension and branching. So obviously this seems important, right? This progranulin is tied to so many different things, and there's not a world where I could study all of them. But I am worried about where low progranulin promotes neuroinflammation. That's sort of what my lab is focused on, and why we decided that progranulin would be important to study. So, to connect everything I've said so far, progranulin protein regulates lysosome health and function. And we know that lysosomes are really instrumental in detecting the onset of an infection and facilitating an immune response. And inflammation is part of this immune response. And then we have this inflammaging, this chronic inflammation that seems to contribute to neurodegeneration. We know that progranulin protein is highly expressed in lysosomes. So to put that all together, my objective over time has become to better understand the role of progranulin in lysosomal health and how this goes on to contribute to inflammation and neurodegeneration. All the pieces seem to fit together somehow. We just don't know exactly how. And so to study this, I use a model of cells. I'm not really into the live animals or tissue or anything like that, but I do a lot of cell culture. So what I use are mouse embryonic fibroblasts. This is a spindle-shaped cell harvested from a mouse embryo. And it's a really, really stable cell line, and it has a really rapid growth rate. So if I'm growing these in a flask, I usually have to split them or maintain them 2 to 3 times a week so that they don't overgrow. They grow really fast. And you might be wondering, why am I not using microglia? I just told you that's so concentrated in microglia. Why in the world would I choose these cells and not those? And the big idea behind that is if I'm trying to understand a biochemical pathway right now, which is what I'm doing, I don't need to use a translational model like microglia. So this is just sort of what I do is try to get the background information. Microglia would be used when I'm trying to do more translational research, basically. So I use MEFs. They're super stable. Like I said, they're very easy. They can undergo a lot of different changes in their conditions, which is really good. So I can try a lot of different things on them, and they'll still survive, and I'll still see results. So the red that you see in this image is tubulin, and that just gives like the cell structure, the outline of it basically. And then the blue is DAPI. That only stains the nucleus of cells. So it just helps you get a cell count. So that's what this image is. It's just MEFs. They haven't been treated with anything, but to give you a good idea of what they look like. They're also very large, so they're very easy to look at. So it's great to take images of and great to use for some of the different assays that I do. So when I'm using MEFs, the 2 probes that I'm focused on are LAMP1 and BMV109. First, BMV109. So this is a membrane permeable fluorescent probe. So it can just automatically go into a cell. And it only fluoresces when it's degraded by cathepsins within the lysosome. So this probe is very cool. It's also very expensive. So I can't waste it, but it's really, really neat to use for some of these assays because it's broken down by these cathepsins in the lysosome, and cathepsins are lysosomal proteases, and they're important for tons of different lysosome processes, one of these being triggering this immune response. So by using this probe, I can look at cathepsin activity in wild type versus granulin knockout MEFs. The other probe that let me backtrack a little bit. I also use baflamycin A1 when I'm treating cells with BMV109. Baflamycin is just to create a control. So all of the cells will get treated with BMV109, and then for my control groups, I'll co-treat with baflamycin A1. And this is just used to prevent lysosomal reacidification. So basically, the cathepsins can't degrade BMV109 when it's in the lysosome, and the BMV109 can't fluoresce. So just to serve as a control group. And then I'm also looking at lysosomal associated membrane protein 1 as a probe. Basically, all that does is give me a lysosome count. So it's an antibody that I treat the cells with, and it helps me see lysosome levels. So which type of cells has more or less lysosome levels, how have they changed when it undergoes different treatments, things like that. So BMV109 and LAMP1 in MEFs. And then there's two different assays that I mostly focus on. The first of these being Western blotting, which I showed you a little bit about earlier, and I'll show you more in a bit. But Western blotting is a way to look at protein concentrations. And this is a really, really brief overview of the process. It takes 2 days to do a Western blot. It's very time consuming, but it's also really, really useful information. And so to do a Western blot experiment, I'll start with my cells that I've plated at whatever concentration I chose in a plate like the one in step 1. And that's when they'll get treated with BMV109. So all the control groups will get baflomycin put on top of the cells. It'll sit for 2 hours. I'll come back and I'll put BMV109 on every single group. It'll sit for 1 hour. And then I start the process of lysing the cells, which lysing the cells basically makes the cells burst, where I can isolate the protein and separate everything else out. So that's what you see in step 2. This protein suspension is at the top. I should only have protein floating around in there, and everything else at the bottom is the rest of the cell parts that I don't care about anymore. And then from there, I do something called a BCA. So this is just a way to check protein concentration to really make sure I actually do have protein that I've isolated, and see how much I'm loading into each sample. So I know if I don't have any, if I have way too much, anything like that. It's just important to do a BCA first. And that's when we get to really starting the Western blot, this SDS page in step 3. So that's loading these samples into a gel. And it's literally like, it's a square like this big, and it's a little bit thicker than Jell-O. But it's this gel that you load this protein sample into, and then you connect it to electricity, basically, and the protein runs down this gel. And you can see it because it has dye in it at this point. So you see the proteins running down this gel, and the smaller proteins move a lot faster than the larger proteins. And so at the end of this run, basically you've separated all the proteins by size. Any protein you had in that cell, they're now separated out across this gel by size. Your smallest ones at the bottom because they move the fastest, your largest ones at the top. Then you go into step 4, which is an electrotransfer. So once you've separated all the proteins on a gel, that's not really useful to you. You can't do a lot with the gel. So you transfer it to this membrane, and it's a small, it's like a piece of paper basically, and it's about this big, and it's really, really sensitive. You can't touch it even with gloved hands. You can only pick it up with forceps, because any time that you touch it, smudge it, anything like that, that'll appear in your images later, and you won't be able to see your results at all. So you have to be really, really careful with this membrane. But you do this electrotransfer, which literally transfers the proteins from the gel on just straight onto a paper underneath them. And then you take that paper, and that's what you use for the rest of the Western blot. So this membrane, then you do the antibody probing step. So I've already treated with BMV109 because that's not an antibody, it's just a probe. But LAMP1 is an antibody. So I do this antibody probing step where I'll treat with LAMP1, and it just basically, it's this antibody that gets dumped right on top of this paper, and this paper soaks it in overnight. I just leave it overnight. It's got my LAMP1 antibody on it. And then the next day, I need a secondary antibody, which just helps make sure LAMP1 was the only thing that was binding. So I treat with a secondary antibody for an hour, and then I'm able to image it. So it's a really long process, lots of steps, lots of places to mess up. But what you get at the end is very, very useful. The other type of assay that I do is immunocytochemistry, or ICC for short. And this process gets really beautiful pictures. The other process, you just get those bands. It's kind of ugly to look at, but it's important. This one is cool to me. So the first thing that you do, again, we're starting with live cells in a plate, and I administer my probe, so BMV109, same thing as always. The only thing that's different here is at the bottom of each of the wells in this plate, there's cover slips. So it's really, really small pieces of glass. They're like this big. And they're going to get transferred to a slide later, because that's what I'm ultimately making, is just slides with cells on them that I can take pictures of. So I administer the probe on top of this cover slip, and I'm still dealing with live cells. But then I fix the cells with 4% PFA or paraformaldehyde and also apply a perm buffer. So this basically sort of breaks down the structure of the cell. It makes them permeable so that anything I treat them with, which later on is going to be an antibody, the LAMP1 antibody, it can enter the cell, but it allows the cell to maintain its structure. So since my end goal is to take pictures of the cell, I don't need it to fall apart. So the other one in the Western blot, I like break it down immediately. I don't care about it. I just want protein. This one, I need the cells to stay together so I can get good pictures of them. So that's why we fix the cells with PFA and then apply this perm buffer. And then you go on to the antibody probing step. But this is all still happening in a plate with the cells in this plate with the cover slips under them. So all happening in a cell culture hood, all very sterile, all of those things. So I probe with my LAMP1 antibody, same as in a Western blot. So you do your primary antibody, then you do your secondary antibody. And then because I'm taking images on a microscope, I also do a fifth step to counterstain with DAPI, which I showed you guys a little bit earlier. That's what just stains the nucleus. So it just helps you see how many cells you actually have. So counterstain with DAPI, and then you get the cover slips out and you put them on a slide. That part is very difficult because if the cover slips are really, really fragile, it's a very small, thin piece of glass. It's super, super easy to break them. And it's really a bummer if you do, because then you have to start all of this over. So you get the slides onto a cover slip and you basically just glue them down and then you can take images. So that's very cool. And that's what I spend most of my time doing. And now I can show you guys some results from that. So the first thing I have here, this is a Western blot. So this is sort of everything you get out of a Western blot. And the first thing I looked at with my MEFs was BMV 109. So what I was able to show is that the signal is strongest in granulin knockout MEFs. And I was able to show this in a Western blot. And this is important. I should have said this earlier. So it's important that I do both a Western blot and immunocytochemistry because a Western blot is quantifiable, so I can get actual numbers out of this. Immunocytochemistry is not. You're just taking pictures, and it's really cool, and they're really beautiful, and they're great to look at, and that's important. But it's also really important that I have numbers. So that's why I need to do both of these. So at the top, you're seeing the results from the blot itself. So this is after I put my primary and secondary antibodies on. The BMV 109 has been sitting on these cells, all of those things, and this is what you get out of it. So the first two lanes right here, you see there's no bands here. If there was BMV 109 signal, so cathepsin activity, because that's what it's showing me, it would show up right here. And there's nothing in the wild type lanes. But in the MEFs that don't have granulin, so my granulin knockout model, you're seeing these two bands here, which is really interesting. And then the last four rows, so my wild type with baflomycin, my knockout with baflomycin, those are controls, and there's no signal here. So that just means that the control did what it was supposed to do and diminished that signal. So it definitely looks like just off that image that there's a lot more BMV 109 signal from granulin knockout MEFs. Which means there's a lot more cathepsin activity happening in granulin knockout MEFs versus wild type MEFs. And it'd be really great if that was the end of it and I could just look at that and assume that, but you have to analyze this. And that's why I have this at the bottom. This is total protein. So this is an entire membrane. This is a cut membrane. So I just cut the slice that I know I'm interested in because that's where BMV 109 shows up at this like specific molecular weight. So I cut that because that's the only part I care about. But before I do that, I scan the whole blot. And this is what's called a total protein. And it shows the protein concentration. Every single type of protein that's on that membrane shows up right here with this stain. And that's important because when I go to analyze this blot, I need to normalize it. So basically lanes that have more protein than other lanes, I need to account for that. So I'm not just assuming like, oh, do I see more signal in the granulin knockout MEFs because they just had more protein in general. So that's why you normalize it to total protein. Sorry about that. So that's why you normalize to total protein. And then once you do that, basically what you get is this graph I have over on the right. So you can definitely see numerically that there was a lot more BMV109 signal, so a lot more cathepsin activity in the granulin knockout MEFs. So after I do the Western blot, I try to confirm this in ICC, immunocytochemistry, and that's what I'm about to show you here. So again, the red is going to be tubulin, so to give you the structure of the cell. The blue is DAPI, which gives you the nucleus count, and then the white is BMV109. So I used 15,000 cells per well. I used a 24 well plate. And the first thing I looked at was the wild type MEFs with BMV109. And you'll see it's a little bit difficult to see because then that's a good thing that there's not a lot of BMV109 signal in these wild type MEFs that were treated with BMV109. But not surprisingly, in the granulin knockout MEFs, it's everywhere, which was like really cool to see. It's super bright. You can see all the little white specks. That's BMV109. So definitely visibly, there's a lot more BMV109 signal in these granulin knockout MEFs. And then again, I have my control groups with baflomycin, and all of the sort of like this white dot here, things like that, that's just background. That basically means the signal is so low from BMV109 that the microscope makes it up. So that's just, that's not actual like BMV109 signal. So the controls worked. There's not a lot of BMV109 signal in the wild type, and there's a ton of it showing up in the granulin knockout MEFs. So this confirms what I found in the Western blot. So after doing BMV109, it was time to move on to look at LAMP1, because I need to make sure that this difference in cathepsin activity isn't just because there's a difference in lysosome number. So basically, is there more cathepsin activity in the granulin knockout MEFs because there's just more cathepsins because they just have more lysosomes? I don't know. So I need to make sure, and that's why I'm looking at LAMP1. So this is a LAMP1 Western blot. And what it shows ultimately is that the wild type MEFs actually have more lysosomes. So whether this is a good or a bad thing, I don't really know yet. But the important thing is this difference in cathepsin activity, this difference in BMV109 signal, was not just a result of a difference in lysosome number. So that definitely means something funky is happening with these cathepsins in this granulin knockout model. So again, with the Western blot, you see this really dark signal in the wild type. You also see signal in the granulin knockout, which is important. Obviously, these cells should definitely still have lysosomes. But this blot, it's not as easy to look at and be like, okay, definitely there's more signal in these, or definitely there's more signal in these. And that's again why we have to analyze these blots. We normalize them to total protein. And then I get the graph on the right, and it does show numerically that there's more LAMP1 signal in the wild type MEFs. So what I can conclude from this, and the immunocytochemistry, unfortunately, I don't have pictures of the LAMP1 because it's been very difficult to get that to show up in images. I'm doing a little bit of troubleshooting, which is definitely a huge part of research. So I don't have results to show you, like actual images to show you today, but that's where I'm at with this in terms of Western blotting. So what I can conclude from all of that, I've sort of already touched on, but just to sum it back up, is that there's definitely more BMV109 activity in the granulin knockout MEFs. So this does confirm that this loss of progranulin is altering cathepsin expression. And maybe the best sort of hypothesis I have for it right now is that it's a compensatory mechanism. So in these granulin knockout MEFs, they seem to have less lysosomes, and maybe this is making the cathepsins work overtime. They're working a lot harder to make up for these less lysosomes to help the cell still do what it needs to do, and part of this is triggering an immune response. And then I've also confirmed that there's more LAMP1 activity in wild type MEFs. So this means that this increase in BMV109, or this increase in cathepsin activity, is not just due to increased lysosomes in granulin knockout MEFs. So that's several years worth of work in my undergraduate career, but now it's leading to a next step that's really exciting, which is in-tap progranulin. So basically, once I finished all of that, it was like, okay, what's next? Where does this lead? What does this mean? I actually don't know. There's so many different things I can do with this, but what I'm focused on right now is intact progranulin. So this is a plasmid, and it's in a viral vector that I can put into cells and make them produce progranulin. So what I've been working on all summer is basically making my own cell line of wild-type MEFs that I transfect with this plasmid so that they produce progranulin. So ultimately, all the time, I basically have progranulin that I can purify and do whatever I want with. And why would I want to do that? Well, with this progranulin that I've purified and I can just hold on to, I can give this back to these granulin knockout MEFs and see what they do with it. What will these assays look like now that they've taken up progranulin? These knockout MEFs that didn't have granulin in the first place, if I give it back to them, what are they going to do? Is this going to make the assays look like the wild type MEFs? Is something else going to happen? Who knows? I don't know yet. I'm still working on making the cell line so I can get my progranulin. But that's the idea, is that I'll transfect just MEFs, make a cell line that I maintain over time that get to produce progranulin, and they'll basically just spit it out into media. So I can collect the media off the top of the cells, and in there is progranulin, and then I can purify it and I can give it to other cells. So that's my plan right now, is to get a cell line that produces progranulin that I'm able to purify and then give back to my granulin knockouts and see what happens with these assays. So I'm very much looking forward to working on that over the next year. And this image at the bottom, this is just what the plasmid looks like. So yeah, with that, I would like to acknowledge my lab. Of course, Dr. Tansey, who has fostered this amazing research environment for me, and I absolutely would not be here without her. Dr. Wallings, Dr. Jowers, Gian and Jake and Drew, Jana and Hannah and Jenny and Noelle, and of course everybody on the right. A lot of this research was funded by an internship I did last summer, so I also need to thank them. That was the SNP program at UF. And then with that, I can take any questions that you guys have.
I hope I'm not the only one lost with this, but, and I have a background, I'm a dentist, but the lysosome, I had to look it up real quickly. You didn't really define, I don't think. The lysosome is a subcellular particle, organelle, I should say. And there's many of them in a cell. How many? Do they vary? And you're talking about going, to put it in perspective, what you're doing, there's a lot of little things up there. I don't think the microphone's on. Can you turn the...
I can hear it. I can hear it.
Okay.
Yeah.
Okay. Doesn't sound like... But what you explain on the cellular level, what you're doing. We see a lot of things up there we're not familiar with, of course. And your lab is, I'm sure. But I don't understand the protein, which was the name of this thing. I forgot it started with
Progranulin, yeah.
Yeah, a little more about progranulin and the organelle. And are we talking about when you're going, are you breaking the subcell, the organ, what did I call it?
An organelle.
The organelle's membrane layer, which is in the cell layer, of course.
Yeah.
We just need to, I think, see a little more of the physical mechanism.
Yeah. So when you think about a cell, just try, I don't know how much like science background you guys have, right? But a cell, just visualize one cell, just a circle, a blob, however you see it. And within that cell, sort of like within our bodies, we have all of these organs. We can't see, we don't know what half of them are doing, but it's the same thing in a cell. They have organelles, and one of these organelles is the lysosome. So if you just, what is the function of an organ? So organelles serve all different types of functions, sort of like all of our organs serve different functions. So you have your heart that helps pump your blood, and you have your kidneys and your liver. Just like that, cells have all sorts of different organelles. So they have like the endoplasmic reticulum, they have a nucleus, and then they have a lysosome. They have mitochondria. So all these different things. If you compare it to our human body, it's like us having different organs, cells have their different organelles. And I'm focused on the lysosome again, because the lysosome serves a lot of processes. Some people refer to it as like the dumpster of the cell, like it just helps get rid of excess like materials, things we don't need anymore, helps process them, break them down, keep the cell clean and happy. But the lysosome serves multiple functions, just like a lot of your organs serve multiple functions. So one of the things the lysosomes does is help facilitate an immune response. And an important thing I think about research, right, and I feel like you hear this a lot about PhDs, is you become an expert in one thing. And so that's why with this research, like it's super specific. I'm going into the cell, I'm picking an organelle. From the organelle, I'm picking one function that it serves. From the one function that it serves, I'm picking one protein that helps it serve its function. And then I'm picking one function that this protein serves and focusing on that. So it's diving through all these layers to pick out one thing, because there's no possible way that I could study all of the different things the lysosome does, all of the different things that progranulin does, sort of like that. So it's very, very specific. But like I said, in the lysosome, one of its functions is to help facilitate an immune response. And progranulin seems to help do this because it's associated with cathepsins, which that's what BMV 109 signal is showing me is cathepsin activity. And cathepsins, they're just an even further breakdown of the lysosome. So you have your cell, you have your lysosome. Within the lysosome, you have cathepsins, and cathepsins have progranulin protein. And these cathepsins help the lysosome carry out this immune response function.
Do you have a hypothesis, though, that could be, of course, tested, a hypothesis, to how it really does this? What does the cell do to do an immune response? In other words, some people, obviously there's all kinds of diseases now. That's the big catchword, autoimmune. How does this happen in theory? I mean, what's your hypothesis on this and your people, I mean? And I mean, that's a practical thing. I know we hate to be practical. We don't have much longer. We got to be practical here.
I think that's a good space for me to, I'm not super like I can talk about progranulin all day, right? But this is sort of stepping out of what I can speak on. But what I can say is what my lab's whole idea, we only focus on neurodegeneration. So all these different neurodegenerative diseases, but there's something, this specifically applies to Parkinson's, but I think it can be applied to other neurodegenerative disorders. And this is what we call the perfect storm. So it's a combination of 3 different factors that throughout your lifetime, and some people combine and make this perfect storm that leads to neurodegenerative disorders like Parkinson's disease. And this is a mix of genes, so literally just what you're born with. There's nothing anyone can do about it. It's just what you're born with. Environmental factors, so some that have been looked at in neurodegenerative diseases are like pesticides, things that you eat, so your gut health. Yeah, your gut health is super, super important. And then the third factor that makes this perfect storm is the inflammaging that I talked about. So this chronic inflammation that happens over time. And all that is basically throughout your life, like we go through so much wear and tear. Like, you know, you're born and you've experienced nothing. And then as you age, like you experience just so many different things. It's as easy as spending time in the sun, right? This puts wear and tear on your body. And so your body undergoes this chronic inflammation as you age. Even if it's not noticeable to you, it's happening. And so the combination of these 3 things is what we think is leading to these neurodegenerative disorders. But my lab we're worried about the genes, right? Because we're also worried, I guess, about environmental factors. Like we know there are some pesticides that are like, okay, we really don't want to be around this, or that your gut health like definitely seems to matter. But my lab is worried about genes, and so there's so many different genes. There's so many that are implicated in neurodegenerative diseases. My gene is granulin. That's what I look at. We have other people in the lab that look at the LRRK2 gene. So LRRK2 is really, really implicated in Parkinson's disease. We have people looking at alpha-synuclein. That's a protein that's encoded by a gene that's associated with these neurodegenerative disorders. So it's all of these different factors and all of these different genes, and it's literally just the perfect combination of everything that we think is what leads to neurodegeneration. But the thing, like I talked about in the beginning, my lab, we're not really concerned with like finding therapeutics right now. We want to know, how can you prevent this from happening? And that's actually where some people in the lab look at gut health, right? Because with Parkinson's disease, for example, 20 to 30 years before you start, like you can really get a Parkinson's diagnosis, people are showing like symptoms of IBS. So if you're showing like these bowel symptoms 20, 30 years, that has to do with your gut health. Are there things we could have done to help maintain your gut health so this basically stopped the IBS from developing, so then Parkinson's won't develop because then you didn't, so it's sort of like catching it before it even begins. Can we do that? We don't know, but we're trying to. So that's sort of a big picture, I guess, if that answers your question. Thank you.
I think you just answered my question. I was going to ask but I'll just say that group, I mean, you're terrific. It was a great presentation, and I can imagine a cocktail party with you and all those people. Oh my God. Wow.
I was going to ask, how far away are the studies from preventing?
I think that's another question that I'm not necessarily too qualified to answer, because like progranulin, it sort of depends on what you're looking at. Progranulin is a very long ways away. Like this molecule is incredibly difficult to understand, and that's partly because it serves so many functions and the lysosome serves so many functions. So if we're trying to isolate one, that's very difficult. But then you have other things like, I guess a big one that a lot of people in my lab also study is tau. So tau is another protein, and in Parkinson's disease, you have tauopathies, which are like tangles of these proteins. And so people studying tau, they've come much further than we've come with progranulin. So things studying like tau, LRRK2, alpha-synuclein, people look at those a lot more than they look at progranulin, so those are a lot closer. It's also sort of this idea of the perfect storm that I talked about is some people don't believe it, right? So like my lab's on the side of like, yes, we think there's this perfect storm, this combination of these three things, but then there's another side of the field that's like, no, I don't think that's it. And so they don't study that, and they don't really care about it. And that's totally okay. These are all just hypotheses. So in the area of neuroscience that does care about it and does believe it and does study it, then you have to think about, okay, what part of it, how are they studying? How far have they gone with that? Things like that. So I can definitely say progranulin needs a lot of work. There's not a lot of people that study it, and it's very difficult to study. But then other things like alpha-synuclein and tau, there's a lot more hope in those areas because a lot more people have studied it and they understand it much, much better.
Thank you. Go ahead.
You've gotten a very interesting job here as an undergraduate. How did you get it? Did you apply for this job? And how many people like you have jobs like this at University of Florida, undergraduates?
Yeah, so the way that I got it, and this is the way that they teach a lot of undergraduates at UF to find research, is just cold email researchers. Like it helps if you, like I knew I wanted to study neuroscience. So I literally just Googled neuroscience staff at UF. And I read through all these researchers and I read some papers. And as a freshman, I didn't understand what any of it meant. I was like, this sounds cool, but I don't know if this is cool or not. I don't even know what this means. And I actually emailed Dr. Tanzi on accident, and I sent her an email that didn't even have her name on it. It was meant for somebody else. It was a big mistake. And it was this whole email I'd written for this other researcher I was interested in. And I was like, look, like I read your paper. I thought this was neat. Like, can we meet and talk about your research and then see if you possibly have any jobs for undergraduates? And I somehow accidentally sent it to Dr. Tansey, and she got it and she was like, I don't think this is for me, but I'm looking for undergraduates. Like, do you want to meet? And so as a freshman, like when you're given an opportunity like that, you don't say no. So I'm like, oh, of course, let's meet, let's talk about what you do. And I met with her and I met with 3 other labs. And I love the way she talked about undergraduate research, because undergrads in the lab are like, we're the bottom of the food chain. Like, we do all the dirty work, like the stuff nobody wants to do. And it's stuff that's important and somebody has to do it. But she talked about me as an equal, whereas other labs I looked at, they were like, oh, you'll never even go in that room. Like, you're too young, too inexperienced. And I mean, that's true. There's so many things that I don't need to be doing that I don't know how to do. But they didn't give me the same attitude of like, we'll teach you how to do this that Dr. Tansey did. So that's why I joined her lab. And for the first year, I washed a lot of dishes. Literally just like washed glassware. And I had an amazing mentor, Cody Keating. He was there the first year and then he's moved on from this lab, but he's the one who first got me into cell culture and progranulin, because that's what he did. So we started really small and he just let me like watch him do some things. And then he sort of handed the project off to me as he left. And so I'm really, really lucky, because a lot of undergraduates don't get the chance to do this much like of their own project. But because the way Dr. Tansey treats undergraduates, because of the mentor I had when I was a freshman, this project just sort of fell in my lap and I've been able to work on it. And she really treats me like her grad students. So she gives me a lot of freedom. She, gives me the guidance I need, but she also like wants me to learn to do things on my own. So that's been really important. And that's how I've been able to do what I've been able to do. And there's tons of other undergrads at UF who are in labs and they go through the same experience. And then there's obviously a huge pre-med population at UF who they do research because it's what you're supposed to do to get into medical school. And so they just like hop on and they'll like dissect rats in a lab for a year and like say that's research. So there's a little bit of a mix. It's the people that really love it and then the people that are just doing it because they're pre-med. But there's also all sorts of levels. Some people get into labs where they don't value undergrads and they wash dishes for all 4 years. Or then there's labs like mine where Dr. Tansey like just is so fantastic to me and lets me really do science, and sort of has pushed me, not intentionally, but now she's the reason I want to get a PhD, because she's given me this opportunity. So
You're paying for this?
Am I paying for this?
Are you paid for this work?
I am, yes. A lot of volunteers, or a lot of undergraduate research start as volunteers, which is what I did. You don't expect to get paid. But when COVID started, and we had to go home for like the spring semester of my freshman year, they could only bring me back if I was an employee. And that's when I started doing real work anyways, like not just volunteer work. So now it is an actual job.
Yeah, the question was, are you paid? I'm not sure everybody heard the question.
Yes.
Yep. I had a comment and question. So thank you so much for being here. I learned a lot today, many words I hadn't heard before. And I also just wanted to comment on just the amount of command you have over the work that you do is more than I've seen from a lot of grad students that I know. So that you should be very proud of that.
Thank you.
Yeah, I'm excited to see just what you do in the future. And the question I had was, so you work with mouse models essentially. Has the lab worked with other types of animal models, or are other ones seen as good analogs?
Not at UF. Like we, in this position, we only work with rats and mice. And I don't even like specifically do that. I had my first experience. Well, I grew up on a farm. So I thought like like I grew up on a pig farm, like my dad's vet, whole circle of life. Like I've seen pigs from start to finish, like they start as babies and they end up in the freezer. So I didn't think working with a mouse was going to be a big deal for me, like dissecting it and everything. But last summer was my first experience and I was not into it. Like the live mouse, I couldn't do it. I had such a hard time. We have, people that do projects where they're injecting a mouse like once a week with stuff. That's not what I can do. So we have people working with mice and rats, and we have people doing like live studies. We have people that dissect them for brain tissue. And then you have people like me who I only work with cells that I get out of a freezer. So I don't even like dissect the mouse and get the cells. I just I have frozen aliquots basically of these cells. I know previously, I don't know, I can't speak on what she did with these, but I know that one person in our lab, it was before she was in Dr. Tanzi's lab, she did work with chimpanzees in Atlanta. So it was neuroscience research, but with chimpanzees, which I think is really cool. I don't think it was really this sort of area of study, but it was still neuroscience research with chimpanzees. But right now, in this sort of thing, all the work you're seeing is with mice and rats. And we also I forgot about this till just now, we have someone in our lab who does Drosophila, so flies. And so he's doing really cool work with flies, but that's not as common. Most of what you're seeing in this area is just mice and rats.
The Zoom people didn't hear that. We had a comment.
Yeah, no, Chipley, Florida. So the panhandle of Florida, 3 hours north of here, the only part of Florida in a different time zone. But yeah, that is how I grew up. So I don't know where I came across neuroscience and not staying on the farm, but.
I hope you get a bachelor's degree and a master's degree at the same time. Is that possible? You're really doing great work.
Thank you. It is where I'm at. It's not possible, but I am hoping to just go straight into a PhD program next fall. So I'll graduate in the spring and start in a PhD program next fall. And ideally, it'll be in Dr. Tanzi's lab. So I basically just get to keep working on this throughout a PhD, and eventually I'll defend a PhD thesis based off of this work.
Thank you.
Thank you.
I think if she's on it, she's going to cure Parkinson's in 4 years. So thank you for everything today. What a wonderful presentation.
Yeah, thank you guys. Thank you for having me.